Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Glioblastoma.
Yeah.
It's a heavy diagnosis, isn't it? The most common, really aggressive brain tumor in adults.
It is, and despite everything we throw at it, surgery, radiation, chemotherapy, it often comes back. Very resistant.
Exactly. Which is why finding completely new ways to tackle it is just so critical.
Absolutely. And that's where some really fascinating new research comes in, looking, right inside the cancer cells themselves.
At their internal clocks.
Precisely. There's this experimental compound, and it seems to target the glioblastoma cell's own internal timing mechanism.
Okay, interesting. And we're basing this deep dive on what? There's a news report?
Yeah, a health news article that summarizes things, but also, crucially, the actual scientific paper that details all the lab work, the preclinical stuff, and even a very early human trial.
Right. So our mission here is really to unpack this for you. We want to understand how this new compound actually works, why it might be a big deal, and what happens next.
Let's get into it.
Okay, so first things first. These internal clocks. You mentioned circadian clock proteins.
Yeah.
What are they doing normally?
Right. So think about your body's daily schedule. Your sleep-wake cycle is the obvious one, but it happens right down at the cellular level, too.
Okay.
These circadian clock proteins, they're like the tiny managers inside each cell, regulating all sorts of daily rhythms when cells divide, repair damage, all that.
Got it. So they keep everything running on time, basically.
Exactly. But here's the twist. This research shows glioblastoma cells have figured out how to, hijack these proteins.
Hijack them? How?
They essentially reprogram the clock machinery to fuel their own relentless growth and replication. It's like they turn the cell's own schedule against itself.
Wow. Okay, so they co-opt the system.
Precisely. It gives them a growth advantage. And that's where the idea comes in. If we can somehow mess with that hijacked clock,
We might be able to slow the cancer down.
that's the hope. And this experimental compound, it's called SHP-1705, is designed to do just that. It's described as a cryptochrome activator.
Cryptochrome activator. Okay, break that down for us. What does activating cryptochrome do?
So cryptochromes, or CRY, proteins are part of the normal clockwork. SHP-1705 specifically boosts the activity of one type, cryptochrome 2, or CRY2.
And what does CRY2 normally do?
These CRY proteins, CRY2 included, act like natural brakes on the cell's clock machinery. When they're active, they slow down those daily cycles.
Ah, okay. So SHP-1705 hits the brakes harder by activating more CRY2.
Exactly. It ramps up the activity of this natural inhibitor.
And why is that particularly effective against glioblastoma?
This is the really interesting part. It turns out glioblastoma cells tend to have unusually low levels of this specific protein, CRY2.
Oh, so the natural brake is already weak in these cancer cells.
Right, which means when SHP-1705 comes in and boosts the little CRY2 they do have, it has a much bigger impact on slowing their clock down compared to healthy brain cells, which generally have plenty of active CRY2 already.
So it's potentially more targeted towards the cancer cells because of their specific deficiency.
That seems to be the mechanism, yes. It exploits a vulnerability.
That sounds promising. So they must have tested this thoroughly in the lab first. Tell us about those preclinical studies.
Oh, absolutely. Extensive work. They started with cells in dishes, and a big focus was on what are called glioblastoma stem cells.
Right. Those are the really tricky ones, aren't they? The ones thought to drive the tumor's growth and recurrence.
Exactly. They're notoriously hard to kill with standard treatments. So the researchers tested if SHP-1705 could actually neutralize these stem cells.
Yeah. What did they find?
The results were quite encouraging. SHP-1705 did impair the survival of these glioblastoma stem cells, and importantly, it seemed to have much less effect on healthy brain cells tested alongside them.
Okay. That's good selectivity.
Yes. And they also compared it to some other clock-targeting compounds that had been looked at before.
And how did it stack up?
It appeared to be more effective, more potent in these tests.
What about resistance? That's a huge issue with glioblastoma, especially when it comes back after chemotherapy like temozolomide.
A crucial question. They tested SHP-1705 on glioblastoma stem cell lines that were known to be sensitive to temozolomide, but also on lines that were resistant.
And did it work on the resistant ones?
Yes. Impressively, it showed effectiveness against both types. That suggests it might have potential even when standard chemo has stopped working.
That could be really significant for patients facing recurrence.
It really could. Dealing with resistance is a major hurdle.
So after the cell studies, they moved to animal models. Mice.
Correct. They looked at mice with human glioblastoma tumors. They found that higher doses of SHP-1705 slowed down the tumor growth.
And did it help the mice live longer?
Yes, it extended survival in those models. And another interesting finding,
What was that?
it seemed to make the tumors more sensitive to radiation treatment when used together.
Oh, wow. So it might not just work alone, but could boost existing therapies like radiation?
That's the suggestion from the mouse studies, yes. A potential synergy there.
And speaking of synergy, wasn't there something about combining it with another experimental drug?
You're right. They did explore that. They paired SHP-1705 with a different compound called SR2-9065.
And what does that one do?
It targets another part of the clock machinery, something called REV-ERB. It was developed by collaborators.
Okay, so hitting the clock from two different angles.
Exactly. And in several of the tests, that combination actually worked even better than SHP-1705 did on its own.
That points towards potential combination therapies down the line.
It definitely does. Often hitting a cancer pathway from multiple points is more effective and can help prevent resistance. The clock is complex, so a multi-pronged attack makes sense.
Okay, so the lab work, the animal studies, they all sound pretty positive. But the big leap is always into humans, right? The phase 1 trial.
The critical step, absolutely. So Synchronicity Pharma, the company involved, ran a phase 1 trial. This involved 54 healthy volunteers.
Healthy volunteers. So this wasn't about treating the cancer yet.
No, not at this stage. Phase 1 is all about safety. Can humans tolerate this compound? What are the side effects? What's the right dose range? You have to establish that first.
Right. First, do no harm. So what were the results? Was it safe?
The findings were good on that front. SHP-1705 was found to be safe and generally well tolerated in these healthy individuals.
Any side effects noted?
The main ones reported were pretty mild things like headache and some nausea, nothing overly serious, which is what you hope for in phase 1.
Okay, that's a really important hurdle cleared then.
Yeah.
So pulling all this together, the lab work, the safety trial, what are the big implications here for glioblastoma treatment?
Well, first, it really puts the spotlight on the circadian clock itself as a potential Achilles heel for this cancer. It's a new type of target.
A different way in.
Exactly. And second, the fact that SHP-1705 is the first clock-targeting drug of this kind to actually complete a phase 1 safety trial is significant. It shows this approach is potentially viable in humans.
And as you mentioned, it seems to hit those glioblastoma stem cells.
Yes, the preclinical data suggests it can target those stem cells, which, as we said, are a major reason current treatments often fail in the long run and the tumors come back.
Plus, the potential effectiveness against chemo-resistant tumors.
That's another key point.
Yeah.
And the possibility of enhancing radiation therapy.
Right. And potentially working in combination with other drugs, like that SR compound.
Yes, the potential for synergy is exciting. And one more practical point.
What's that?
SHP-1705 is taken as a pill, an oral medication.
That makes difference for patients, doesn't it? Easier than infusions, especially if they're already getting radiation or other treatments.
It definitely could reduce the treatment burden, which is always a consideration.
So lots of potential angles. And you mentioned the clock's role might be even broader, something about immune suppression.
Yeah, there's emerging evidence suggesting these hijacked clock proteins might also play roles in things like suppressing the immune system's ability to fight the tumor and helping the tumor build its own blood supply. Targeting the clock might disrupt those processes too.
So lots of promise, but we need to be realistic. What are the limitations of where the research stands right now?
Absolutely crucial to remember. Most of the really compelling data we've discussed is still preclinical. It's from cells in the lab and from animal models.
Okay.
And while the phase 1 trial showed safety in healthy people, it didn't tell us anything about whether the drug actually works against glioblastoma in patients.
Right. Safe doesn't automatically equal effective.
Exactly. That's the next big question. Does SHP-1705 provide a real clinical benefit for people with glioblastoma? We don't know that yet.
So what's needed to find that out?
The essential next step is a phase 2 clinical trial. This would involve giving the drug to actual glioblastoma patients to see if it shrinks tumors, slows progression, or improves survival. That's where we find out if it truly works.
That phase 2 trial will be key then. Now, we also have some insights from the researchers themselves, right? what did Dr. Steve Kay say? He was the senior author.
Yes. Dr. K really emphasized that there's mounting evidence that these clock proteins get, well, co-opted by the brain cancer stem cells to fuel their growth. He stressed the potential of targeting the clock to stop them multiplying.
Makes sense.
Oh.
And Dr. Priscilla Chan, involved in supporting the research, what was her perspective?
Dr. Chen focused on that critical gap in current care, the failure to really get rid of those glioblastoma stem cells. She expressed hope that SHP-1705 might help fill that gap, leading to more lasting treatment effects.
Addressing the recurrence problem head-on.
Exactly.
And did Dr. K compare SHP-1705 to earlier attempts?
He did. He mentioned that compared to some previous compounds his team had looked at, this one, SHP-1705, seems to be more potent and just better suited for tackling glioblastoma specifically. So represents progress in their drug development efforts.
Okay, so it really does feel like a potential step forward in a field that desperately needs it. For you listening, having gone through this with us, what are the key things to maybe watch for or think about?
Well, first, definitely keep an eye out for news about that phase 2 clinical trial. That's going to be the big test of efficacy for SHP-1705 in patients.
Right, that's the crucial next update.
Second, if this area interests you, maybe read a bit more about glioblastoma itself. Understanding the challenges really highlights why new approaches like this are so important.
Good point. Context is everything.
And finally, maybe just reflect on how amazing and complex these basic cellular processes like the internal clock are. They're vital for health, but as we see here, they can also be exploited by disease. It really shows the potential of therapies that target these fundamental mechanisms.
So wrapping up our deep dive today,
Right.
we've looked at this novel compound, SHP-1705. It targets the internal circadian clock that glioblastoma cells seem to hijack.
Preclinical studies show promise hitting stem cells, working against resistant cells, maybe boosting radiation. And importantly, it passed initial safety tests in humans.
But, and it's a big but, we need that phase 2 trial to see if it truly works in patients. Still, it represents a genuinely new strategy, a hopeful direction against a really tough cancer.
It really does. It highlights how understanding the deep biology of cancer can open up completely new avenues for treatment. It's a testament to ongoing scientific discovery, definitely offering a glimmer of hope.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.